Hello, good evening. Today's topic is the subtopic in centrifugation, that is density gradient centrifugation. Till yesterday, we have discussed about the basic principle of centrifugation and sedimentation, types of sedimentation uh, instruments, that is preparative centrifuge and analytical centrifuge. Then we have talked about different types of rotor designs, the concept of wall effect and how it is rectified when we go for zonal type of centrifuges rotors. Then we have started discussing on the factors which contribute to the sedimentation process and the efficiency of the sedimentation. We have calculated, rather derived different formulas which talk about how to calculate the F centrifugal field, G, that is relative centrifugal field, RCF and RPM concepts, time required for centrifugation, and the factors which are going to contribute while deciding on selection of a particular type of rotor. Right, all these things we have discussed. Also, we have discussed differential centrifugation in the previous lecture. So today's topic is on density gradient centrifugation. Yes, it was as early as in 1950s. Scientist Breck had not Breck, B-R-E-A-K, Brack mm, uh, described about this particular technique. So when it comes to density gradient centrifugation, yes, it is ultra centrifugation. It's a really a powerful technique for fractionation of mixtures of cells, a mixture of particles, mixture of components of a cell, that is subcellular organelles, and even for purification of isolation of macromolecules. So here you are going to have separation on the basis of size and mass of the particular molecule of your interest. You are going to have a column of liquid, that is the centrifuge tube, in which you are going to develop a gradient, solution gradient. What is gradient stands for? That is concentrations are going to change. So at the bottom of the tube, you have highest concentration. At the top of the tube, you have lowest concentration of the material that you have chosen to develop the gradient. The materials which are generally chosen, they include glucose, glycerol, or ficol, then cesium chloride, and so on and so forth. So we'll be talking about these various chemicals which are used. Of course, it is not limited to glucose or cesium chloride, but ficol, then sodium metrizoate, cesium chloride के साथ sodium metrozeate के साथ you have certain molecules which have certain brand names, Ludox, right, or back extractor, like that, which are developed by different companies, and you have those ready to use density gradients for the process of your research, sorry, process under research. So when it comes to mm the technique, yes, it has lot many applications, right from cell organelles to macromolecules to virus purification, purification of molecular biology components like DNA, RNA, types of DNA, right? Then food microbiology and clinical microbiology for isolation and purification of pathogenic bacteria if they are there in the sample, so on and so forth. Right, so this is overview of the technique. Now coming to the details. Uh, Here, when you talk about the word gradient, it tells you that there is going to be a change in the concentration, and you have a tube in which all these concentrations are available for separation purpose, for sedimentation purpose. So you can imagine a tube, centrifuge tube that we have been seeing in previous lectures. Also, we have seen how they look like, right? Centrifuges and centrifuge tubes like this, right? You have either narrow bottom one or the circular bottom. That is how they differ, and the depending on what is the purpose of or use or application of the particular centrifugation that you are doing, you have to choose the appropriate tube that we know now. Now the concern is when such tubes are filled with gradient material, maybe sucrose, maybe ficol, maybe cesium chloride, they of course the selection of the gradient material also is important. But at the same time, when we say gradient, there are 2 points. Either it is a steep gradient or it is a continuous linear gradient. So when I say step gradient, that means you are going to layer the solutions of different density in a centrifuge tube. So if I talk about, say, in this case on right hand side, if you see the table, if you see the slide, you have 50% of sucrose, then 40% of sucrose, 20% of sucrose. Like that, the concentration are reducing, right? So you are going to layer 50% sucrose at the bottom. It is then you are going to layer 40% on the top of that. On the top of 40% sucrose, you are going to layer the volume of 20% sucrose, and like that you can go on reducing up to 5% sucrose solution, which is going to be at the top. So you are going to layer solutions of different density, different concentrations, right, one above the other. And the last step is at the top, which is going to be the layer of solution which is less dense, right? So this is step gradient. And when you say continuous or linear density gradient, that means you don't have such zones. This is the zone or layer of 40%, this is a layer of 30%, 20%. No, not like that. The density is going to change smoothly and continuously. So it is either going to be linear or exponential. That is one thing. But from very low extreme to very high extreme, but there are not zones as such which can be separately or distinctly separated. So you are going to have a smooth and continuous increase or decrease in the concentration, top to bottom and bottom to top. That is how it will change, right? So continuous gradients can be produced from the step gradients by allowing sufficient time to diffuse. Can you imagine you are having a tube at the bottom of which you have put 50% sucrose, and at the top of which consecutively you are adding 40%, then 30%, then layer of 20%, then layer of 10%, and layer of 5%. How long these layers would remain separate as steps or as separate zones? For some specified time, provided you are storing it under low temperature, say 4 degrees Celsius, such separate zones can be maintained. But if you allow these zones to diffuse, right, if you give sufficient time for the diffusion, then 50% sucrose, and at the top of that you have 40%. So 50 and 40 ka jo boundary hai, there you will have some degree of mixing, and it would generate 50, 49, 45, and then 40. That is how a continuous gradient would generate. So you have to allow diffusion of these layers, so that you are going to hold the tube after step gradient is prepared. You are going to hold it for longer time and then allow the diffusion to occur. Then you can expect that linear gradient is generated due to diffusion of molecules in the centrifuge tube. Now, here is a list of different materials that are used and the purpose for which they are applied. For example, in the column 1, you see dextran, picol, glycerol, percol, sucrose, bovine serum albumin also as the material to develop the gradient. Now, if you talk about any of these are non-ionic solutions. These are not salts like cesium chloride, right, or KBr or NaBr, sodium bromide or potassium bromide. They are also used, but here it is not the case. They are all are non-ionic, as you see in the second column. Third column says about maximum density at 20 degrees Celsius. So this is the factor that tells you about the density of the particular material that you have chosen. Now, based on the desired molecule or the molecule of your interest to be separated, you have to choose the best possible gradient material wherein these values also of concern. But you see there are not much differences. It ranges between 1.13 to 1.3035. That's it. But when it comes to centrifugation, this 0.01 unit difference also is very important. Now, why the column number 4 is there? Ultraviolet absorbance. So this is the material which is holding, which is held in the tube and which is having the material or particle to be separated. When the separation is going on, are you able to monitor the separation process? Are you able to scan what is actually happening? Yes, that is possible by attaching other analytical instruments to the centrifuge. And one of them is UV scanner that you can attach. So it is very required that your material should not absorb UV. right? The gradient material should not absorb UV, then and then only the molecule under study would be characterized by UV based analysis, right? Now, when I talk about dextran, it is used for microsome banding and wholesale separation. Picol is commonly used for wholesales, subcellular components, as well as for viral fractionation. When it is to glycerol, generally it is used for membrane fragments and protein separations. See the stringency. Then comes percol, again wholesale and subcellular component. Sucrose is used for viruses, membranes, proteins, subcellular particles. Wide application sucrose has got, and it is most commonly adopted system, you can say. So these are the examples of different gradient materials that are used. Now, how to prepare the gradient? For example, we take the example of sucrose, the most commonly used sucrose gradient. Now here it is prepared in a centrifuge tube. It can be done manually or it can be done by taking help of automated machines. So here you have a gradient maker and a peristaltic pump. The pump is to push the particular sample into the holder tube. Now what is the sample here? The sample here is nothing but the 5% sucrose solution, 20%. Are you able to see these two tubes? One is labeled as 5%, another is labeled as 20%. These are the two tubes which are holding sucrose solutions, right? Now this is the pump, peristaltic pump, which is going to uh, pressure out or by pressure pump out the solutions in 5% and solution in 20%. The solution is going to get pumped out and is going to fill the centrifuge tube to develop the gradient. All right. So a capillary is connected to a tube from the peristaltic pump, and it is put at the bottom of the centrifuge tube. And what you are going to do is you are going to apply the pressure. So gradient will be generated because part of 5% and part of 20% will get added to it. So if you want to mix the two in particular proportions so that desired gradient you can prepare, or 20 at the base followed by 5 at the top, and then allow the diffusion to happen so that may you reach to gradient between 5 and 20. That is possible, but yes, different concentrations can be mixed to get 15%, 10%, and like that. Fine. So sample is to be loaded once your gradient is ready. So this is how a peristaltic pump and a gradient maker instrument will look like. Now comes the next point, that is uh, the continuous gradient preparation, right? Now here you have a certain instrument which is from a particular company I have shown here, which is showing you the details like, yes, what is the company, what is the brand, right? And here are the two tubes which are going to hold the sucrose solutions of different concentrations. Here is the outlet pipe, here is the microcapillary attached, and here is the tube which is going to be used as a gradient tube, right? So here the gradient flow is controlled by a valve which is centered between the two chambers. Here is the valve, right? The outflow channel is fitted with a adapter which is can be used with any needle, and of course it's like a tap, so you can switch it or switch on or off, and then the solution will come down through it. So one gradient can be prepared in some about 10 to 15 minutes, and a ready gradient will be available to you. Now let's watch some steps for preparation of the gradient, right? Just watch carefully. Step 1, tearing the gradients. Fine. You have to make the weight of each tube. Centrifuge is balanced, filter is balanced. Sample volumes are to be fixed. So this is the ice basket in which your tubes are kept. Here we are taking the dimensions. You have to add a sample, which is roughly 2 millimeter of the space. These are the working details. Need not remember. It is just to know about. Yes, the tube, inner diameter, the volume it holds, maximum sample of the volume, minimum sample of the volume. All these parameters you have to take into consideration. Now here they are using the tip. It is cut for the easy delivery of the solution. The lid is open, right? They are taking the weight of the tube, right? So for all the tubes, it is like that, right? It is important to layer the exact same amount of sample on each tube, right? That is very important. See how carefully the sample is getting loaded on the top of the gradient so that it slowly spreads over the surface of the liquid column, the gradient column, right? Yeah, a very uniform layer. And you can very well see the sharp interface of the two layers, right? So this is just a overview. One more thing is to be shared, but at the end of the lecture. Now here, when we talk about gradient, uh, uh, you have different materials, as I said. But when it comes to gradient centrifugation, it is been divided into types. Basically, isopycnic gradient centrifugation and red zonal sedimentation. And further, it is equilibrium zonal sedimentation. The little advanced um, method you can say, right? Now, in which the density gradient can be used. That's it. So, when centrifugation continues until all the particles in the gradient have reached a position where their density is equal to the medium, and this type of centrifugation separate different particles based on their different densities. Sucrose alone may not provide sufficient density for isopycnic banding of many viruses. So example is you can use it for a separation of viruses, but again you have to choose a appropriate gradient. But what is the point? The particles are going to get at particular location where the density of the particle matches with the density of the liquid at that position, and it is going to form a band. Now when it comes to red zonal one, the sample is layered over the preformed gradient, and each particle again is going to sediment as a zone or band to the gradient at a rate depending on its size, shape, and density. And the centrifugation is stopped while the particles are still sedimenting. So there are two basic minor differences. We will talk about it in the next slide. But yes, an equilibrium zonal sedimentation is like red zonal sedimentation only, except that sedimentation is continued until most of the particles have reached an approximate isopycnic position. So, at what time the centrifuge will be stopped? That is going to differ when you talk about rotational and equilibrium zonal sedimentation. So, equilibrium zonal sedimentation will end after sedimentation is completed, and centrifugation is stopped while particles are still sedimenting in red zonal. So these are the two basic differences. Now, when it comes to type of rotors, so high speed preparative ultracentrifuge, which may be with swinging out type of a rotor or fixed angle rotor can be used. Now, when you are going to separate the I mean, allow the sedimentation to go, the bands are separated. You can visualize them through light scattering. So you can get to know that yes, centrifugation assembly along with the density gradient prepared, that is one part of the analysis. Second part is identification or scanning, you can say, of the separated materials using different instruments like light scattering, light absorption, UV absorption also we have seen in the previous slide. So any of these or even in microscopes, electron microscopes, um, sampling is immediately done and you are going to get the scanning electron microscopes can give you the details about it. Now, once the centrifugation is over, the bottom of the density gradient tube, centrifuge tube is punctured and the contents are allowed to drip into a series of sample tubes and fractionating device based on upward displacement of the contents. Part by part, from the base where it is punctured, you will collect the fractions and they will be going for analysis. Now here is the schematic representation. Now let's talk about red zonal in details first and then isopycnic. Red zonal separation of particles is based upon the characteristics of material that is under separation, and it includes size, shape and density. And of course these factors, as we know, are going to be dependent on density and viscosity of the medium and the applied centrifugal field. Now what is happening? The sample solution is carefully layered. So you have prepared the gradient first and the sample is loaded on the top of it, and then you are going for centrifugation at ultra high speed. Right. And when the particles which are getting centrifuged through it will find the density that matches with the density of the particle will stop migrating from there onwards. It will get sedimented there. So small density particles at the top and heavy large density particles at the bottom. That is how separation takes place. Now, this technique has been used for separation of enzymes, RNA, RNA DNA hybrids, subcellular organelles, etc. Now, the density of the particle is greater than the density of the gradient, and if centrifugation is continued, now they are separated here. And what is the second case we are talking? Density of the particle is greater than the density of the medium. What will happen? So, if you are continuing the centrifugation, all the particles will end up settling at the bottom of the tube. Centrifugation time also is very crucial, and the gradient that you are choosing, whether it will be 5 to 20, 5 to 25, 5 to 27% sucrose, that needs to be optimized, right? So the point is, if the density of the particle is greater than the density of the gradient and centrifugation continued longer, pellet will get formed and not the separated zones, right? As against red zonal, here if you see in isopycnic, here is the sample is shaded throughout the gradient, right? In the previous case, we had layered it on the top of the gradient. Here it is different story. It is mixed along with the entire length of the tube. And as centrifugation is applied, again the particles will get separated based on the density. Yes, where the particle matches, where the density of the particle matches with the density of the medium, a band will form. But what was the starting point? All of them were mixed throughout the length of the tube. There where it differs in the red zonal and isopycnic. Now the particles migrate through the solvent gradient until they reach the point where ion density matches and they form the zone. So it is used to separate the particles on the basis of their pion density, which is independent of time. In previous case, time was important. Second factor to compare. Now, each particle sediments to a position in the centrifuge tube where the gradient density matches with the density of the particle. Right. Now, even if after that sedimentation continues, right, centrifugation continues, there is not going to be any more separation which is going to occur. How long you may continue the separation, centrifugation, further sedimentation will not take place. The particle is buoyant at the position and remains at that particular position only. So, when I say buoyant, that means the density of the medium is more than the density of the particle. So, banding is not exactly at the density where it matches, but it is lighter than that. The particle is lighter than the density of the medium at that particular point. So buoyancy is there, and here it will keep on floating. It will not sediment, it will not migrate upwards. So particles differing in density but have the same size can be separated by this method. Subcellular organelles like Golgi apparatus, peroxisomes, mitochondria, etc. you can separate. So in the first tube, what you see is a homogeneous distribution of sample and the gradient in the tube. as you apply the centrifugal force, see the arrow indicated here, and this is how you are going to get the fractionation of the components, right, under the influence of centrifugal force when you are running the centrifugation in a gradient. The gradient gets redistributed and band is formed at their isotonic positions. If the density of the gradient medium must be higher than the density of the particle to be separated, that is a key point to be remember. Now, when it comes to applications, we have seen that sucrose can be utilized. Fine, sucrose is utilized, but uh, here, I am sharing one paper with you, sucrose gradient sedimentation analysis of mitochondrial ribosomes. What are mitochondrial ribosomes? You have ribosomes outside the mitochondria which are involved in total protein synthesis of the cell. But mitochondria have some set of ribosomes which are involved in synthesis of certain selective proteins which are essential for oxidative phosphorylation process which occurs in the mitochondria, right? And if you want to separate this particular fraction, you can take help of this particular density gradient centrifugation. Now, what is the great thing in that? Point is, sedimentation will depend on centrifugal force applied and density and viscosity of the gradient. Importantly, it will also depend on size, shape, density of microsomes, micro micro my is to mitochondria and a ribosome. So my to bosomes. That is how it is spelled. Particles present on the sample. Variation of the technique found coupled with additional downstream approaches analyze process of uh, mitoribosomes biogenesis and the composition assembly intermediates or monitor the interaction between extra-ribosomal proteins with individual mitoribosome subunits or monosomes. So here not only separation, but you can find out the interactions also if they are happening between extra-ribosomal proteins and mitoribosomes. Now, this is the process given and the manual details. It is just for extra reference that yes, not only for separation, but you can use it for comparison of non-interacting and interacting components, which are having different origin, different location, different function. So that is one part. And second thing which I wanted to highlight here is you can profile whatever material you are separating by gradient. You can prepare the gradient. So gradient maker, here are the details of the site which you can choose for knowing about the gradient maker for more details and immunoblotting. Further, these sections can be taken for immunoblotting and analysis. Right. Now here comes another paper which talks about one-step centrifugation, which is commonly used for separation of blood cells, and Ficol gradient is commonly used. In this method, after centrifugation, the peripheral blood mononuclear cells are located on the top. So if you are centrifuging blood and Ficol gradient is used, so you are going to have the peripheral mononuclear cells on the top of the separation fluid, and other blood cells like erythrocytes and granulocytes will sediment at the bottom. But here they have done one additional step that they had taken 75% of lymphocyte suspension and it was separated using one step density gradient centrifugation. So what they had used heparin, sodium heparin blood, right? So heparin is anticoagulant and sucrose is the gradient material and they had used 10% to 100% sucrose gradient they had prepared by mixing various quantities of sugar and the milky water that was prepared, and 4 ml of diluted blood was layered on the 4 ml of each sucrose solution and it was centrifuged. So 10% ka gradient tha, 20 ka tha, 30 ka tha. That is how they had done for 45 minutes at 1000 rpm. And what they observed, when it was layered on 40% sucrose, the peripheral mononuclear cells which were forming a top layer only in the previous centrifugation process, here the separated PBMS were analyzed and they showed 75% lymphocytes, 23% monocytes, and 2% of other cells. So, use of sucrose gradient only, but what concentration would be better for good separation needs to be optimized. Right. Here is another point to be talked about. When you talk about density gradient centrifugation, yes, like blood components, viruses, we have talked about. Bacteria can be recovered from soil using sucrose gradients. Different types of protozoa can be separated from river water, feces, etc. Gradient centrifugation in Percol can be used for distinguishing different populations of pathogenic bacteria and to separate live and dead cells. Right? Buoyancy will be differing, so you can use it for that. To a certain extent, this technique can be used to separate microbes from food and separation of bacteria from milk and yogurt, etc. Density gradient, ultracentrifugation, yes, we have talked about subcellular component separation and purification. So mitochondria, peroxisomes, endosomes, and exosomes also can be extracted from high viscosity samples like saliva. So viscosity of the medium from which the source from which they are extracted also is going to contribute a factor that will decide the efficiency. Now this is another one. OptiPrep is one lab in USA which is making use of a non-ionic iodizanol. So this is another molecule which is used as a gradient material, and 1.32 gram per ml for isolation and purification of various types of particles, including exosomes. So when you talk about any of these density gradient materials and density gradient centrifugation, the use is that it is very simple, high speed, good separation, and intact organisms or intact materials without having any damage to the material that is under separation. You can run the process. That is very important. At the same time, you can remove the compounds that might interfere or inhibit the detection methods. So ability to remove inhibiting or interfering material present in food, that can be evaluated by further techniques. That means when you do centrifugation by density gradient, all other unwanted contaminating materials will get removed. For example, you are extracting DNA from the pathogens which are present in the food, and that DNA you are trying to separate. So if there are any fragments of the DNA which are coming from any other source and are undesired, they will not form band at a particular density which will match with the target molecule. And how do I rely ki only pure molecule is coming and it is not contaminated? Yes, it can be assured or it can be checked after you are going to analyze these products by subsequent methods, like you can go for PCR and check, or you can go for nucleic acid sequencing based on amplification, or you can go for ATP measurements. So purity of the desired molecule or complete good isolation is assured by density gradient centrifugation. But there are certain limitations. For example, if you take the separation of exosomes, and you want a higher purity, just excuse me for a minute, it got disconnected somehow. Yes. So you are talking about higher purity, then you have to talk about enrichment. Maintaining the structure of the molecule also is very important, because ionic materials can affect the structure of the material under study. Osmotic environment is very important. So sucrose, cesium chloride, all these things are going to affect the biological activity or structure of the particle or subcellular organelle under study. So you have to be very careful in using the gradient and selecting the gradient. Selection of gradient material, advantages and limitations when we talk. Fractionation of human serum protein, lipoproteins, role of salt and sucrose. I want to share one paper on this. I'll go to the next, right? Not this. Right. Cesium chloride density gradient for, no, not this. This one. This is for bacteria. Where has it gone? Okay, fine. It is mixed somewhere. Anyway, but the point is here, role of salt and sucrose. I'll get this paper next time when we meet. All right. So application part or related to density gradient, we will discuss in the next discussion. Right now, you can just tell me if you are any what we say questions and queries related to the discussion we had till now. 3 minutes are there, so I am trying to share one more video with you. Till then, you can ask if you have any query in the points discussed. All right, I'm sharing that video with you right now. Making a gradient, yes. I shall start from here. How to make a gradient, right? Is it seen? Yes. Gather the necessary items. Then test tubes, tube caps, marker blocks, lubrication, silicone tubes. And now let's observe the activities. So if you're going above 50%, then what you should do? The gradient having, right, it is fixed in the tube, in the inner wall of the tube. With a plunger, you are fixing it. So in another tube, right, it's in the similar manner. So here, yes. So no air bubble is present. We are releasing the content in the syringe. 10% because you are layering in the tube, right?